Today we're going to be talking about Watson and Crick's DNA model. The general structure that they proposed was a deoxyribose sugar bonded to a phosphate group, while that sugar was also attached to a nitrogenous base. Hydrogen bonds stabilize the DNA molecule throughout, but we will talk about those later as well. The sugar also has a carbon ring, whereas the 1' carbon attaches to the nitrogenous base right here. The 2' carbon distinguishes the DNA molecule from the RNA molecule by only having an H bonded to it, whereas the RNA molecule only has an OH. The 3' carbon bonds next to the 5' carbon, of which a phosphate group is bonded, where you can see over here. Talking about the strands, there are major and minor grooves, which allow proteins to bind/recognize DNA from outside the helix during processes such as transcription, where that would need to be a thing. Watson and Crick also realized that the strands would need to be anti-parallel. And all that means is that one strand would lead from the 3' to 5' in one direction, and in the same direction, the other strand would lead from the 5' to 3'. Now, these strands are also either able to be left-handed or right-handed, but the DNA strand that is the most accepted model is right-handed, as seen down below. And all that means is that it twists in the right direction. So we have to talk about the nitrogenous bases. We have adenine, guanine, cytosine, and thymine. The first two can be sorted into the purine category, just accounting for their double ring structure, while the latter two can be sorted into the pyrimidine structure, which accounts for their single ring nature. So these nitrogenous bases, due to hydrogen bonding, pair up, and also they stack upon each other in common forms of DNA, so they can enable the DNA molecule to be more sturdy. So DNA itself is a negatively charged molecule because of, if we go back here, the phosphate grouping. This phosphate group has a negative charge, but if we place that molecule into a high salt concentration buffer, that negative charge is neutralized, which means that because in two strands of DNA, the phosphate groups push apart the molecule, if we neutralize those, the molecule will be more stable. So the temperature of melting, which is almost the halfway point to a completely denatured molecule of DNA, is increased because that pushing apart of those same charges is neutralized by the high salt concentration. So this is my citations. Thank you for watching.